Xstrahl hosted a virtual meeting, Advancements in Translational Radiation Research, featuring a panel of leading radiation researchers highlighting translational research done over the last 10 years using the Small Animal Radiation Research platform. The meeting featured 6 leading radiation researchers who highlighted some of their research outcomes using SARRP. Dr. Anthony Chalmers, Chair of Clinical Oncology of the Institute of Cancer Science at the University of Glasgow, summarizes research done to optimize radiation therapy delivery.
To get a little bit more sophisticated, we wanted to look at whether we could optimize irradiation of our GBM models further. And so this is where Alistair Rutherford's work came in, looking at some simple beam arrangements as I've described, but also some ARC arrangements either through the gantry or couch. So Alistair was able to define the tumour and the organs at risk, including the eyes, the brainstem, the mouth, contralateral brain and ipsilateral brain, and did some nice dosimetry studies. This is the parallel opposed that you've seen already. So the tumour coverage is in blue and you can see it's very good. And we have, with this model, irradiation of a portion of the ipsilateral and the contralateral brain. With the superior beam, we can completely eliminate the contralateral brain as described. And then with the gantry arcs, we have a variety of different scenarios. So with the gantry arc, we can get good dose distributions, but this is probably not reflective of the clinical scenario when we tend to irradiate the tumour and a margin around that. Whereas the couch arcs give us a plan that's much more representative of a clinical plan. And so if you are interested in looking at immune effects in the vicinity of the tumour, you may wish to use this kind of system where you're getting a more clinically relevant dose distribution. It did come at the slight expense of dose to the contralateral brain, but those are marginal effects. And you can see with different angles, slightly different DVHs as well. So just comparing them all, really, they were all very safe to give, very good tumour coverage in all of them, differential effects on ipsilateral and contralateral brain. A couple of them affected the brainstem, but these are very low percentages here, around 10%. And I was pleased that none of them really gave significant dose to the mouth.
For more information on the Small Animal Radiation Research platform, or to learn how SARRP can help you with your research needs, visit xstrahl.com/SARRP.